Hello, everyone. We are group number 10. My name is Henka Eichelberger.
I'm Klista Gilger.
I'm Shelby Little.
And I'm Brennan McManus.
And our question was, what is the role of the S protein in attachment and entry in the coronavirus life cycle? We are also going to include changes to the structure of the S protein during binding and uptake. So the coronavirus spike protein mediates the attachment and entry of viral RNA into the host cell. The spike protein is formed by 3 monomers, which are each made up of 2 subunits, S1 and S2, as labeled on this slide. The S1 subunit is a globular head-like subunit, and its role is to bind to the ACE2 receptor commonly found on the epithelial cell surface in tissues in the throat and lungs. Hence why SARS-CoV-2 and previous SARS viruses affect the respiratory system.
The important regions of the S1 subunit include the receptor binding domain, which also includes the receptor binding motif that actually makes contact with the ACE2 receptor. This region has a high affinity for the ACE2 receptors and therefore will bind with the receptor upon close contact. The next important region of the S protein is the S1-S2 furin cleavage site. This region is an arginine-rich region with the sequence RRAR, highlighted in green, which gets cleaved when the S1 subunit is bound to the ACE2 receptor. Surrounding this cleavage sequence, we can find a high concentration of serines, which are often post-translationally modified via O-glycosidic linkage. This motif serves as a mini mucin patch, which serves as a sort of barrier to protect the S1-ACE2 interaction, as well as the fusion peptide on the S2 subunit.
This brings us to the S2 subunit, which is more fibrous in structure and plays a role in the fusion of the viral membrane with the host cellular membrane. When S1 binds to the receptor and the furin cleavage site is cleaved, the S2 subunit undergoes a drastic hinge-like conformational change. The first region of the S1 subunit is the fusion peptide, which includes a crack sequence that has high affinity towards cholesterol, which can be found within a cellular membrane. This segment of S2 binds to the cholesterol within the membrane, anchoring the S2 subunit to the host cell. There's also a large loop, followed by an aromatic region. The aromatic region interacts with the hydrophobic portion of the cell wall, acting as an anchor similar to the fusion peptide.
Therefore, altogether, when the fusion peptide binds, the loop allows the subunit to have a hinge-like conformation, bringing the aromatic region close enough to also bind to the membrane. This process brings the virus membrane close enough for the host and viral membranes to fuse, and ultimately allows for the genetic information, the positive strand RNA, to get into the host cell, hijack the cell's machinery, and begin replication. Thank you so much for listening.